welcome to today's class. Today we are going to study about the process translation under the subject Biochemistry BP 203T. In the previous lectures we have studied about RNA transcription and DNA replication. Now we are going to study about translation. So, what is translation? Translation is a process when we are biosynthesizing a protein or a polypeptide in a living cell is referred as translation. Translation is the third step of our central dogma of molecular biology, that is when we are synthesizing proteins after the process of RNA transcription. Then that means we are synthesizing proteins from RNA. This step is called as translation. So in this step the representation of biochemical translation of 4 letters, that is the basic letters of our nucleotide sequence, that is AUGC and ATGC, that is adenine, uracil, guanine, cytosine, and adenine, thymine, guanine and cytosine. In this language we are synthesizing our proteins. So, in the information from nucleotide, that is DNA and RNA, to our 20 letters amino acid. That means we are now changing the letter from our nucleotide sequence, that is ATGC and AUGC, into 20 amino acid languages. We have 20 amino acids, 10 are essential and 10 are non-essential, and proteins are what? Proteins are the polypeptide sequence or the functional polypeptide or the polymers of amino acids, and amino acids are, there are 20 amino acids. So, the protein synthesis may be divided into following steps. First, we need the components for protein synthesis. So, we need to collect all the component. Second is the activation of amino acid. Third is the protein synthesis. Fourth, chaperones and protein folding, which is very important, and the fifth step is protein post-translational modification. That means as transcription we need to do some post-translational modification in this step also. So, let us start with the first part, that is requirement of the components. In this, what we need here, the first very important thing we need here is amino acids. So, for doing the protein synthesis we need several amino acids, and amino acid, there are 20 amino acids in which 10 are essential and 10 are non-essential. The essential amino acid means we need to consume them with the help of our diet. In diet we need to consume the essential amino acid, whereas non-essential amino acids we do not need to take them with our diet. It is synthesized by our body. It is synthesized by various cycles, various synthesizing cycles in our body. So, the non-essential amino acid, our body is able to make those non-essential ones. So, the protein synthesis can occur only when desired amino acid is available in plenty of form to fulfill the protein synthesis. If there is any deficiency of of amino acid happens, protein synthesis will not occur. So, it is a very important step that we need to understand. For doing the protein synthesis, our body require each and every amino acid which is going to be used for the process. If there is any deficiency happens in that amino acid, the process will not fulfill. So, next what we need is ribosomes. Ribosomes are the functionally active centers or the factories where the protein synthesis will occur. So, if there is any kind of problems occur with the ribosomes, again protein synthesis will not occur. So, let us discuss something about ribosomes. Ribosomes, they are consist of the 2 subunits. One subunit is big and one is small. The functional ribosomes have 2 sites also. One is A site and the second is called as P site. Each have their own each of their own functions. Like A site is for binding of amino acyl tRNA, whereas P site is for binding with peptidyl tRNA. That means both the sites are doing different functions. A site is for binding of amino acyl tRNA, whereas P site is for binding of peptidyl tRNA. Next, in case of eukaryote, there is another site. Earlier we have studied A site and P site, that is in case for prokaryotes, and in eukaryotes along with A site and P site, one more site is there, that is called as E site or exit site. These thus the eukaryotic ribosomes contain three sites, A, P and E on the ribosomes. The ribosomes are the cytosomal function of the cell. They are found in association with rough endoplasmic reticulum, which we generally call as RER, to form cluster RER ribosome, where the protein synthesis occurs. That is, now the next thing is polyribosomes. What are those? Polyribosomes are when several ribosomes simultaneously translate on a single mRNA. Then that ribosomes will be called as polyribosomes. That means many ribosomes are there for translation on a single mRNA. Then it will be called as polyribosomes. Next is messenger RNA, that is mRNA. The DNA has passed on the genetic information in the form of codons to mRNA to translate into protein sequence. So, mRNA, as the name suggests, is the messenger RNA. It will transform into codons, genetic informations. It will transform the genetic information into the codon. That means it is sending the message from one place to the other place for the protein synthesis. Next is tRNA. tRNA helps as they transfers the amino acid, the desired amino acid from one place to another place for the protein synthesis. That means they carry the amino acid and hand them over to the growing protein polypeptide nuclear chain in man. There are about 50 different RNAs, whereas in bacteria around 40 tRNAs are found. Then we definitely need energy source for protein synthesis. So, for energy sources we do have ATP and GTP, which are supplying energy for protein synthesis. We need some protein factors. There are the process of translation involves a number of protein factors. These are needed for initiation, elongation and termination for protein synthesis. Second step, now we have all the things together. We do have energy molecules with us, we do have amino acids with us, we do have ribosomes with us, we do have other small things which are like mRNA, rRNA, which are very important, playing very important role in the synthesis of protein synthesis. We do have everything with us. Now let us activate the amino acids. For doing this process, the amino acid should be activated earlier. Like amino acids are activated and attached to tRNA so that they can be sent to the growing protein synthesis. So, for that there are two steps involved. One is the amino acid is first attached to the enzyme utilizing ATP to form enzyme AMP, amino acid complexes. So, when we are attaching our amino acid with the enzyme and ATP, then there will be the formation of AMP, amino acid complex. In the next step, the amino acid is then transferred to the 3' end of the tRNA to form amino acyl tRNA. Finally, when we have activated the amino acid, now we have everything in line with us for the protein synthesis. Now let us start the protein synthesis. In this step, the protein synthesis occurs on the ribosomes because they are the perfect places, perfect centers, factories for the protein synthesis. The mRNA is read here and in the 5'3' direction only. The polypeptide synthesis proceeds from N terminal to C terminal. Now this needs to be understood very carefully. When we study the structure of amino acid, it has an N side and a C side, that is N terminal and C terminal. if we make the structure of a general, if we make the general structure of amino acid, then it has a carbon, so here this is the general structure of our amino acid, in which there is an amino group and a carboxylic group. So the amino group is called as N-terminal, whereas the C group, that is carboxylic group, will be called as C-terminal. So every amino acid has the same structure, only this R group is replaced every time with several alkyl groups. Okay. So the initiation, the protein synthesis occurs on the ribosomes. The mRNA is read in the 5' to 3' direction from N-terminal to C-terminal. It starts from N-terminal to C-terminal. N-terminal is on the left hand side, whereas C-terminal on the right hand side. The prokaryotic mRNA are polycistronic. Since a single mRNA has many coding regions that code for different polypeptides. In contrast, eukaryotic mRNA is monocistronic, since it codes for only single polypeptide. When a mRNA is coding for multiple proteins, multiple amino acids, then it will be called as polycistronic, whereas when it is coding for single polypeptide, it will be called as monocistronic. In case of prokaryote, translation commences before the transcription of gene is completed, whereas in eukaryote organisms, it is in eukaryote organism, as we know, it is a complex organism, since the transcription occurs in the nucleus, whereas transcription takes place in the cytosol. So it is organized in that way, eukaryotes. Whereas in prokaryotes, there is only single organelle, that is nucleus, inside the cell. So there is the transcription, translation starts, whereas transcription is still going on. So in eukaryotes, this is not happening. So this is the initiation process. In initiation process of translation in eukaryote is a little bit complex. It involves at least 10 eukaryotic initiation factors, which are also called as EIFs, eukaryotic initiation factors. Some of the EIFs contain multiple 3 to 8 subunits. So initiation can be of different steps. First step here will be the ATS ribosome dissociates to form 40S and 60S subunits. Here in the first step, the ribosome has to be dissociated. That means the ATS ribosome will be dissociated into two subunits, that is 40S and 60S. Two initiation factors, namely EIF3 and EIF1A, binds to the newly formed 40S subunit and thereby block its reassociation with 60S subunit. That means we need this 40S subunit here, very importantly. A ternary complex containing MET tRNA and EIF2 bound to GTP attaches to 40S ribosomal subunit to form a 43S pre-initiation complex. So now the 40S ribosomes becomes 43S pre-initiation complex. Now let us see how that 43S will become 48. The binding of mRNA to 43S pre-initiation complex results in the formation of 48S initiation complex through the intermediate 43S initiation complex. This, however, involves certain interaction between some of the EIFs and activation of mRNA, and then finally it becomes our 48S initiation complex. This 48 initiation complex then finally binds to the 60S ribosomal complex, which it was dissociated from, to form ATS initiation complex. The binding involves the hydrolysis of GTP which is bound to EIF2. This step is facilitated by involvement of EIF5. So, as we have seen in these steps, there are so many EIF, that is eukaryotic initiation factors are involved. First of all, we dissociate the ATS ribosome into 40 and 60, then the 40S ribosomes subunit will become 43S pre-initiation complex, then it becomes 48S initiation complex, and finally again there is an incorporation of that 60S ribosome with 48S ribosome initiation complex, and finally we got our ATS initiation complex. Now that means the complex is ready for the synthesis process. Now let us initiate the translation in prokaryote. The 30S, that was which we studied here we have studied about eukaryotes. Now we are going to study about prokaryotes. So in prokaryotes, the 30S ribosomal subunit is bind to initiation factor 3IF, that is initiation factor, and attached to ternary complex IF2, formyl met tRNA and GTP IF1 also participates in the formation of pre-initiation complex. A 50S ribosome unit is now bound with the 30S unit, which finally produces 70S initiation complex in prokaryotes. So we know here that prokaryotes has 70S ribosome, whereas eukaryote has 80S, and this 70S is divided into 30 and 50, whereas in the ATS eukaryote is divided into 40 and 60. Now let us discuss about the elongation. So elongation is done like ribosome elongate the polynucleotide chain by sequential addition of amino acids. The amino acid sequence is determined by the order of the codon in the specific mRNA. So mRNA, the role of mRNA here is very important. So it is divided into 3 steps. First is binding of amino acyl tRNA to A site, peptide bond formation. In this peptide bond formation, the peptidyl transferase catalyzes the transformation of peptide bond, and translocation, the ribosome moves to the next codon of the mRNA towards 3' end. So in these 3 steps, the elongation happens. First is binding of amino acyl tRNA to A site, peptidyl bond formation, and finally the translocation. Once we have got the desired amount of length of our polynucleotide chain, yes, then we need to terminate the steps then and there only. So for the termination, one of the stop or terminate signals should be there. So there are 3 signals. Once the translation, the elongation process have any of these 3 signals, it needs to be terminate the step. So what are those? That UAA, UAG and UCA. These are the called as termination signals to the growing polypeptide. A termination codon which act as a stop signal do not have specific tRNA to bound with it. So as soon as the growing polypeptide chain will have this sequence like UAA, UAG or UCA, it terminates the process and we have our full grown polypeptide chain. That means we have our protein synthesized then and there only. As the termination codon occupies the ribosomal A site, the release factor, namely ERF, recognizes the stop signal and it finally stops the complete signal. Now, what are the inhibitors of protein synthesis? Again, we need some inhibitors to stop the protein synthesis in bacteria, viruses only. So, there are certain drugs to used. First is streptomycin. Streptomycin causes misreading of mRNA and interferes with the normal pairing between codons and anticodon. And finally, whenever there will be a misunderstanding or misreading of these codons, there will be no synthesis occurs. Second is tetracycline. Tetracycline inhibit the binding of amino acyl tRNA to ribosomal complex. Then we have chloramphenicol. Chloramphenicol is a competitive inhibitor to the enzyme peptidyl transferase. And lastly, we have erythromycin, which inhibits the translocation by binding with 50 S subunits of bacterial ribosomes. So, these are the agents which finally inhibit the protein synthesis. Now, the fourth step of protein synthesis is chaperones and protein folding. Like we do have in the transcription step, we have studied earlier, in the transcription steps, after the synthesis, after the transcription step, we got our primary transcripts. Primary transcripts are not activated. We need to activate them by doing post-transcriptional modifications. Likewise, here in the translation step, the proteins which are forming, we need to change some of the structure of the protein, some of the folding has to be done for that, so that the proteins which we have synthesized become functional. So, chaperones or chaperones are heat shock proteins. They are originally discovered in response to heat shock only. That is why they are called as heat shock proteins. They facilitate and favor the interaction on the polypeptide surfaces to finally give the specific conformation to the protein. Proteins have four structures, that means primary structure, secondary structure, tertiary structure and quaternary structure. So, to get those structure, chaperones plays a very important role, then only the protein will be functional. Second, the chaperones reversibly bind to the hydrophobic region of unfolded protein to give the folding to the intermediates. That is why chaperones are very important for the protein folding. Next, they can stabilize the intermediates, prevent formation of incorrect intermediates and prevent undesirable interaction with other proteins. All these activities of chaperones help the proteins to attain compact and biologically active conformation. The 3D conformation of proteins is important for their biological functions. Now, the protein, what will happen if there will be protein misfolding? Because there is a high chance when we are folding the protein, there is a high chance of protein misfolding also. So, what is protein misfolding? How it can hamper the activities? Let us discuss about it. Protein misfolding is the failure of protein to fold properly, generally leads to its rapid degradation. That means as soon as the protein misfolds, there will be rapid degradation of that synthesized protein. A disease known as cystic fibrosis. It is a common autosomal recessive disease. Some cases of cystic fibrosis with mutations that result in altered protein have been reported. That means it is so dangerous, protein folding, misfolding is so dangerous that it can cause some autosomal recessive diseases also. Certain neurological diseases which are due to cellular accumulation of aggregates of misfolded proteins or their partially degraded products which have been identified. The term prions are your proteinaceous infectious agents is used to collectively represent them. That means that whole thing will be called as prions. So, protein misfolding if ever happens, that can create so many dangerous diseases, so many dangerous conditions. What are the types of chaperones? There are two types of chaperones, HSP, that is heat shock protein 70, and heat shock protein 40. That means they have two, they are namely HSP 70 system is the mainly consist of HSP 70, that is 70 kilodalton heat shock protein, and HSP 40, 40 kilodalton HSP, that is heat shock protein. And chaperone system, there is a large oligomeric assembly which forms a structure into which the folded proteins are inserted. The chaperone system mainly has HSP 60 and HSP 10, that is 60 kilodalton HSP and 10 kilodalton HSP. So, these chaperones are very important for the correct folding of proteins. That is the final step of our translation, that is post translation, sorry, there is a writing mistake there, post translational modification it should be written, but by mistake it has written post transcription. So, post translational modification, these modification include protein folding, trimming by proteolytic degradation, instant splicing and covalent changes, which are collectively known as post translational modification. Next, proteolytic degradation, some portions of precursors molecule are removed by proteolytic to liberate active proteins, it is referred as trimming. Intene splicing, that is inteins are intervening sequence in certain proteins. Inteins must be removed and exteins ligated in the appropriate order for the protein to become active, and covalent modifications are done by modifications in amino acid in proteins may be converted to active forms like phosphorylation, hydroxylation and on. This is the whole structure which shows the post translational modifications. These are the references which I have referred to. Thank you so much. Have you enjoyed your class?